Beyond Guam, a dark depression opens inside the southern Mariana Trench. This is Sirena Deep, overshadowed by the more famous Challenger Deep. Its floor lies nearly 11 kilometers beneath the restless western Pacific. No sunlight reaches this isolated basin, and no diver sees unaided. The first mystery is not whether it is deep, but how it differs. Sirena occupies another pocket within the same immense subduction trench. Its slopes descend through darkness towards sediment gathered in a narrow hollow. Maps reveal a distinct abyss, yet maps alone cannot explain its character. Every reliable contour comes from sound, traveling through water and returning. At this scale, a tiny timing error can distort an enormous depth. Researchers, therefore, compare repeated tracks before accepting any deepest sounding. What appears smooth from orbit becomes broken terrain under careful sonar. If you descended here, pressure would rise relentlessly with every meter. The unseen basin waits beneath almost 11,000 meters of seawater. Its lesser fame makes the evidence especially important to examine carefully. Sirena Deep belongs to the Mariana Trench, where the Pacific Plate descends. The Pacific Plate bends beneath the smaller Mariana tectonic plate. That downward flexure creates a long trench rather than one simple pit. Separate depressions form where faulting, sediment, and plate geometry vary. Challenger Deep lies farther southwest and contains Earth's accepted deepest point. Sirena Deep occupies the trench's southern sector, east of Challenger Deep. It was once commonly called HMRG Deep after a mapping organization. Modern depth estimates place its bottom close to 10,700 meters. Exact values differ because sound speed changes with temperature, pressure, and salinity. Survey teams correct acoustic travel times using measured water properties. Multibeam sonar sends many sound beams across a swath beneath a ship. The returning echoes reveal slopes, ridges, channels, and relatively level patches. Autonomous and remotely operated vehicles can approach features ships only infer. Their cameras observe sediment, rocks, and animals within a narrow illuminated field. Direct images remain local observations, not portraits of the entire depression. Sirena's water is near freezing, although pressure prevents ordinary ice formation. Pressure at the floor exceeds 1,000 times atmospheric pressure at sea level. Such conditions compress gases and challenge housings, seals, batteries, and electronics. Sediment arrives from sinking particles, slope failures, and material transported laterally. Steep walls can funnel organic debris toward the deepest trench basins. This concentration may create food patches within an otherwise sparse environment. Verified context, therefore, begins with geology, measurement, and uneven resource delivery. The basin's shape records a plate bending, fracturing, and sinking into earth. Normal faults cut the descending plate as its outer edge curves downward. Earthquakes reveal continuing deformation around the trench and deeper subduction zone. However, an earthquake location does not directly describe Sirena's seafloor ecology. Sonar indicates morphology, while cameras test what particular surfaces actually contain. Sediment cores can preserve layers deposited by quiet settling or sudden disturbance. One hypothesis links thick event layers to earthquake-triggered slope failures. Yet, similar deposits may have multiple causes without independent dating evidence. You can picture a tranquil floor, but the geological record may contain abrupt pulses. Chemical measurements show how oxygen and nutrients vary within sediment and water. They cannot identify every organism responsible for a measured reaction. Microbial cells persist in deep sediment, processing buried organic matter slowly. Their activity is inferred from chemistry, genetic material, and controlled incubations. Each method answers a different question and carries a different limitation. Samples also change as they rise through decreasing pressure and warming water. Pressure-retaining instruments reduce that disturbance, but cannot remove every sampling bias. Observed amphipods demonstrate that mobile scavengers reach extreme Mariana depths. They follow chemical cues toward carrion and consume concentrated food rapidly. That action converts a rare food fall into movement, growth, and reproduction. Other small invertebrates live within or across the sediment surface. Their patchy distribution probably follows food, grain size, oxygen, and disturbance. No single landing can establish abundance across Sirena's entire floor. Repeated lander deployments are needed to separate true patterns from chance encounters. Baited cameras attract scavengers, so their images cannot represent undisturbed community structure. Unbaited surveys reveal natural spacing, but may record very few active animals. Together, these observations sharpen knowledge without pretending the darkness is fully sampled. Sirena Deep also tests how life adapts to crushing hydrostatic pressure. Cell membranes must remain functional when pressure favors tightly packed molecular structures. Hadal organisms adjust membrane lipids and stabilize proteins through biochemical mechanisms. These adaptations have energetic costs within cold, food-limited surroundings. When organic particles arrive, microbes release enzymes and begin chemical breakdown. Scavengers detect dissolved cues, travel toward the source, and accelerate consumption. Burrowing animals disturb sediment, changing oxygen penetration and microbial habitats nearby. Those cue-action-consequence sequences connect behavior to trench nutrient cycling. Large food falls may briefly transform a small area of the basin. Between such events, many organisms rely on dispersed particles or buried carbon. Trench geometry can retain material that would spread across flatter abyssal plains. This makes Sirena a natural laboratory for studying topographic concentration of food. If you watched only one baited deployment, abundance could look deceptively high. Longer observations help distinguish temporary gatherings from persistent local populations. Sirena's isolation is relative because currents and sinking material connect surrounding waters. Human-made pollutants can also descend through food webs and settling particles. Their detection at great depth would indicate transport, not a pristine sealed world. Plastic fragments and persistent chemicals require careful identification and contamination controls. Exploration, therefore, carries a responsibility to measure impacts without creating new ones. The basin's geology also matters for understanding hazards along subduction margins. Future surveys must repeat high-resolution sonar tracks across previously mapped terrain. Changes between maps could reveal slope failures, sediment movement, or measurement error. Pressure-capable landers should sample several habitats instead of one convenient flat site. Long-duration sensors could record currents, temperature, chemistry, and particle arrival together. Sediment cores from separated locations could test whether event layers correlate. Genomic evidence may identify metabolic potential, but not prove activity by itself. Direct rate measurements under in situ pressure would strengthen ecological interpretations. You would still see only narrow windows through each camera's illuminated cone. Combining windows across seasons and years can gradually expose broader patterns. Reliable depth claims will continue to depend on calibrated sound speed profiles. Reliable biological claims will depend on replication, controls, and preserved sample context. No instrument can turn one encounter into proof of a trench-wide rule. The most valuable discovery may be how strongly Sirena differs within short distances. Its floor could contain stable patches beside scars of recent disturbance. Future evidence must connect terrain, deposits, chemistry, and organisms at matching scales. Until then, Sirena Deep remains measured but incompletely known beneath the Pacific. The opening dark depression returns as a real, distinct basin whose deepest stories still require direct evidence.